Okay, hello everyone. My name is Ali Al-Shahib. I'm from Public Health England. Has anyone heard of Public Health England? Good. Yes, so we are in the Bioinformatics unit, and today I'll be talking to you about infectious diseases and public health. Before that, I really need to know what your background is. I mean, I was sort of not really given much details about your background. Have you guys done bioinformatics, like a course in bioinformatics or something? Some? Okay, a module. So there's a module in bioinformatics. Okay. Some people have. Right. Okay. Because because I'm assuming in certain areas that bioinformatics is sort of taught. But anyway, if you don't understand something, please do interrupt me. This is, I'm very much a champion of an andragogy approach of teaching. Anyone knows what an andragogy approach is? Compared to a pedagogy? No? Okay, so pedagogy is what's being taught at schools, you know, where a teacher tells the student, this is how it is, yeah. Whereas an andragogy approach is, I think, what is now starting to practice this at universities is where you actually have a dialogue, you have a discussion. It's not just the teacher talking. So hopefully we can do the andragogy approach today. Okay? Good. So Public Health England, just a very quick introduction to PHE. It is part of Department of Health at UK government. It recently was sort of shifted UK to the UK government. It was mostly the NHS before, but there's all these changes that they call the Quango. The different organizations where you gotta do the put the different specialities into the same area, etc. Anyway, there was huge changes, and now we're government with civil servants. This is sort of the formal heading where what is the aim of PHE? We protect and improve the nation's health and well-being, and reduce health inequalities. It sounds all very juicy. So specifically, microbiology services. So in PHE, we have many different areas. We have, I mean, the main areas are, you know, they look at obesity, they look at smoking, and, you know, it's public health and stuff like that. But what we specialize in is in microbiology and infectious diseases. And I come from, not originally, but I come from Colindale, which is, anyone been to Colindale? Yeah, Colindale PHE, that's the main thing it does, it's infectious diseases. But PHE has many centers scattered all over the UK. So in microbiology services, we provide specialist investigations and control of disease outbreaks, chemical incidents, radiation, etc. As I said, in Colindale, we just deal with disease outbreaks and infectious diseases. Yeah, we provide evidence-based science and clinical practice and specialist microbiology. And we support the wider health, public, and NHS hospitals. And we'll get into that. It makes a bit more sense as we progress in today's talk. Okay, so the questions we often ask of a pathogen isolate are the following. So, as you all know, you know, if there was to be an outbreak of whatever, of say in a farm, children visited a farm, and then there was an outbreak. You know, a lot of children then got sick. Then they come to PHE. They say, "What the hell is going on here?" So what we do is we isolate the site. We don't let anyone come in. And what we do is we try to identify, the first thing, we try to identify the isolate. What is it? Which bacteria is this? And And that is the identification stage, where we identify the infectious agent or exclude particular infections and associated risks. Yeah? And then once we identify, say for example, it's E. coli. Yeah, we identify this is an E. coli outbreak. We have a department, a gastro department in PHE, where they start to get involved. And then they'll ask the second question and then say, okay, what characteristics does it have? You know, what is it? Which type of E. coli? As you very well know, you guys know what typing is? Yeah, so in every bacteria, in every bacterial species, there are different types, and types are generated based on their different genomes, right? Their different genetics makeup, and certain mutations. As you progress in evolutions, you get mutations, as you very well know. So that will change the behavior of that particular bacteria. And once it starts to change the behavior of the bacteria, it's still E. coli, for example, but they change the type, right? And so there are various different types of typing systems, mechanisms that scientists use to try to identify a bacteria based on its particular type. Yeah? So I'm going to talk a lot about typing today. This is core to microorganisms and bacterial identification. Yeah? So what characteristics does it have? Of course, this is typing. Is it potentially resistant to antibiotics? You all know the issue of antibiotic resistance now, and it's talked about a lot, and this is really the main thing here. We have many bacteria which they think they're smarter than humans, and so therefore they're what's happening is they're developing genes that are becoming resistant to certain antibiotics. For me, I had a personal journey through this. Last year is exactly actually December 2016. My youngest daughter, she had a urine infection, a bacterial urine infection. And usually when you go to they do an examination and they take a sample, etc. They find out if it's a particular bug, and they quickly give you antibiotics. So they gave her the first-line antibiotics, and she became worse, right? And then they thought, okay, she might be resistant to that particular bacteria. And they gave her another antibiotics, and still she wasn't. So then she was admitted to hospital. She was isolated because they had no idea what's actually going on. So anyway, eventually, after many, many days of suffering in terms of her condition, the results came back. Unfortunately, it takes around 3 days for them to identify the resistance results. So which antibiotics is this particular type of bacteria resistant to, right? So they go through a whole scan of different antibiotics, and they find out which particular antibiotics is sensitive to or is it resistant to in that particular sample. So then the results came out, and then they said, okay, now we know which particular antibiotic we should give, because the results came out and said this particular antibiotic is sensitive to. And then they treated her with that, but then it still wasn't working, and then they went to I think there were around 6 antibiotics they tried at the end. And then the sixth one worked, and then she became better. So this is just an example, a personal example of how serious antibiotic resistance is. And the way that they try to find out if a particular bacteria is resistant is you need to know the type. You need to know the typing. You need to type the particular bacteria to find out which antibiotics is resistant to. And of course, you got to ask, are toxins present? Because then, if our toxins present, and if there is an outbreak, if there is a national outbreak or a local outbreak, etc., and these toxins can kill. And the thing about viruses and bacteria, about microorganisms in general, is that they're unpredictable. They can change very quickly, especially viruses. Viruses, they replicate very quickly, and also They have a high mutation rate, so they can change throughout evolution. And then when a new strain comes up, right, and starts killing people like Ebola, right, everyone starts to panic. They say, oh, okay, this is a new strain. We don't know much about it. So it's going to cause lots of problems, etc. So that is the danger behind these particular organisms. Is that all okay? Who's asleep?
Between the strain and the type?
It's a good question. So a type is, so a strain, you can, in a particular bacteria, you can have multiple strains, okay? And that's based on a type. So if you want if you want a general definition, there is not much difference. But if you want a really detailed definition, if you find a new organism, right, that hasn't been identified, and that's a new strain, okay? But it can have the same type as another already identified strain, okay? So the way you differentiate between different strains is according to if it's not been seen before. But they could still have the same type. I mean, the way they define types are, it's quite a process. It's not just one particular mutation they find and then they say, okay, this is a new type. It all, again, depends on which particular typing mechanism, etc. The thing is that today I was told to speak generally about microorganisms and public health, but you know, the typing world is quite a big one, and I'll go through certain typing mechanisms and how we've, in whole genome sequencing, we've come up with faster ways to type organisms. Okay? Good. The third question is, how does it relate to other isolates? This is vital to know when you have an outbreak. So if you have an outbreak of, say, E. coli in that particular farm, you got to say, okay, have we seen this before? Is this organism known to us before? Have we seen such a thing before? If we've seen this before, then we know about this organism. If we haven't, then we start to panic, because this is a new mutation, a new strain. Yeah. So do cases of an infection have common source, or is there evidence of transmission? What is the source and what's the risk factors? Is it food, school, travel to certain countries? I will give you an example of an interesting outbreak that has happened recently where this applies to. Okay. Answers will help public health and clinical staff to take effective action. So the best ways is it's really treating the affected, protecting others, or limiting further spread. I mean, these are all open questions. I mean, people are still thinking, okay, you know, what is it? Because the thing is that if someone has been infected with a bacteria, before you actually know what bacteria is, you give them antibiotics. That's the first thing you do in a GP, right? You go there and they look at your tonsils, or you might have an ear infection, but they can't find, they wouldn't know which bacteria this is. They'll have a guess. They got to send a sample to the lab, and that will come, the results will come back in like 2-3 days. But they'll quickly give you an antibiotic, right? The thing is that, do you do that? You treat the affected, or what you've got to do is you've got to make sure that you know how to limit the spread of that particular bacteria, especially if it's a bacteria that is unknown. Yeah, so typing, pathogen typing, basically gives a bug a label, a label that you can identify that particular unique species, that unique strain with a label. So if we discover isolates with the same type, we can include or exclude individual cases to an outbreak. This is the advantages of finding out. We can establish an association between an outbreak of food poisoning and a specific food vehicle, like an egg mayo sandwich, for example. You would want to know, if you know the type, you would want to know which, you want to establish this between an outbreak of something like food poisoning or something just of a specific food. You want to also trace the source of contaminants within a manufacturing process. Again, I'll give you an example of an outbreak which then they went and found out the source was an egg, a site in Germany where they store eggs and they distribute eggs and stuff like that, and they found out that was the source of the contaminants. The type also helps to determine changes in microbial populations in response to intervention. So if we have an outbreak and then we start to vaccinate quite a large number of people, we would want to know if that vaccination has helped, right? So again, we want to find out if the population has responded to that particular type of bacteria. So typing helps us to get to know this, study variations and trends in pathogenicity, virulence, and antibiotic resistance, as we have mentioned. So, any questions so far? Okay. So 20th century microbiology. I'm just going to go back in history for a bit, just very briefly. Now, I'm not an expert in this, but as a bioinformatician, by the way, I didn't tell you who I am. I just told you who my name is. You assume someone from the street is just talking rubbish. Well, no, I'm a bioinformatics scientist. I finished my PhD in 2005, so that was many, many years ago. In bioinformatics. And yeah, so I've been working since then, been 9 years in Public Health England, working on different projects and outbreaks and all that. So I don't really get into the lab. We just use computers with the dry people, what they call the wet and dry. Have you heard of the two phrases? The wet and dry. So we're the dry people. But we do the most exciting stuff. The guys in the wet lab, they just do routine stuff, which is a bit boring. Okay, so yeah, has anyone done culture before? Bacterial culture? Yeah, so you know all about that. So again, here, the same thing. We still do culturing. You know, you still got to grow the bacteria. And now this is one of the main things that scientists are really trying to find out. How can we speed up bacterial identification? Because if I was to take a sample from you, I still need to grow the bacteria so I can extract the DNA. Yeah? And that will take how long to grow a bacteria? Sorry? Sorry? Okay, just give me an average. A couple of days. Yeah, usually average a day, 24 hours. Usually at 24 hours you give for a bacteria to grow. And the thing is that this patient might have died by then. You know, if you wait for the bacteria to grow and things. So that's why they treat them with antibiotics straight away, even before knowing which bacteria it is. But still, we need to find out this information. So now what they're doing is they're trying to, there's lots of ways, but they still haven't succeeded to try to directly extract the DNA from the bacteria without actually making it grow in different types of cultures. Yeah? And this is ongoing. There's a technology, if anyone wants to know, there's a technology called nanopore technology, which is trying to do that. You can Google that and find out more information. Yeah, so culture, you know, they still do culture, and they used to do that. And the way to identify, okay, a question is, if I wanted to identify, if I have a piece of my DNA that I'm interested in, right, and I want the sequence of that piece of DNA, how would I do that? Do you guys know what I'm talking about? Put on a sequencer. That's a very good step. But before that, what would you do? Have you heard of PCR? Polymerase chain reaction. Yeah. So that is what you do. You grow the DNA and you develop. We don't want to go into details, but there are PCR machines, and you'll develop the different positions of the sequence, And then you amplify the DNA, and then you get your sequence out. So then, that happens through, you got to do a culture step and stuff like that. So this is what culture is, and again, this is to find out whether a bacteria is a gram-negative or a gram-positive bacteria. They have different identification ways. There is a company called, it's a commercial thing called API strips, which contains around 20 tests in a strip by itself. And then one of the tests, you can differentiate between gram-positive and gram-negative bacteria. So this is done before, and I believe is still being done by certain companies. And you've got also serotyping is done, sensitivity testing for antimicrobial resistance, and you've got phage typing and all that. We'll get into some of these, but these are the typical techniques that are being done now and used to be done. And what is alternative? And of course, you've got the gel-based typing methods where you can identify which particular genes are resistant using gel. So in viruses, I know some people are interested in viruses here. Previously, they used to do crystallography. They used to mostly concentrate on structures to identify. So when they look at a particular structure, which is very expensive doing crystallography and so, they identify a particular virus. And also look at an affected or an uninfected organism. But now, and then they do PCR, amplify that particular DNA, and then draw up a tree where they have these particular so-called clades, where they put together all the different types of viruses together based on their sequences of that particular region of interest, and then they draw a tree. I don't know if there is a... And then they draw a tree based on the identifications. And this is all just based on PCR, okay? Now, transforming 21st century microbiology with whole genome sequencing. Now, what I've realized is that I haven't even given an introduction to whole genome sequencing here, because I've inherited these slides from someone. Okay, what is whole genome sequencing? Sorry, who's talking? Oh, hi. Sorry, say again?
It's not just the exons.
Yes.
The whole thing.
What's the whole thing? The introns, exons, and everything else. It's basically everything. Everything in the genome, right? It's what is called the genome. It's basically all the DNA. You sequence every single thing, right? And that's the technology of that's why it's called whole genome sequencing. Remember, what's the difference between a PCR and a WGS?
EGS is not specific to...
Yes, absolutely. So whole genome sequencing is you sequence the whole genome, whereas a PCR, you have a specific target. Yeah, so you have a gene, you want to amplify that gene and sequence that gene. That's what PCR is all about. But whole genome sequencing is you sequence the whole genome. Now, how much do you think it costs to sequence one bacteria? Take a wild guess. Sorry? Just take a wild guess. I won't judge you, don't worry. £100? 30 pounds? Anyone advancing 100? 150 we have. 150. I'm not selling bacteria, I'm just sequencing. 150. 100 pounds. Okay, this seems like a sort of an average of 100 pounds. It's a good guess, but it's wrong. It's actually someone said 30. I think you said 30, right? Yeah, it's around 25 pounds now to sequence one bacterial genome. But remember, when I say sequence a whole genome, that doesn't mean that I'm going to give you every single thing, because the technology now is that it will leave some gaps. Again, I don't want to go into details into what are anyone know reads? Sequencing reads and stuff. So it depends on the technology now that is used is from a company called Illumina. You've heard of Illumina? Yeah? So Illumina have short reads and they have long reads. Usually the long reads are more expensive, but short read sequences inevitably will leave gaps. And these gaps, if you want to sequence the whole genome, fully complete the whole genome, you've got to do PCR to fill in these gaps. Yeah, to get a whole genome. Anyone heard of GenBank? Yes. What is GenBank? This is an andragogy approach. You're making me do pedagogy. What's GenBank?
Online genome repository.
Sorry?
It's an online genome repository.
Yes, online genome repository. It contains, basically, you do a search, whatever bacteria or whatever species you're interested in. If it's an algae, plant, human, whatever. You type in the name, the scientific name of it, and then there's a good possibility you might get a sequence of that genome. Yeah? But then they'll say if it's a complete genome or if it's not a complete genome. That's the difference. Sometimes they say it's a non-complete genome. If it's a complete genome, it means that they've filled in all the gaps. Yeah? But if it's not a complete genome, then obviously they've sequenced it, but without filling in the gaps. Yeah? Okay. So yeah, that's the whole genome sequencing. It's a technology now that is advancing greatly, really, really much. And a lot of companies now there's lots of startup companies now, and they're crazy for bioinformaticians and genomics experts. And I mean, I get calls from agencies coming and saying, please, can you work for this? And the salaries are very good and stuff like that. So if you're interested in getting into more about bioinformatics and data analysis and what they call data scientists and all that, then there's plenty of job opportunities, because there's lots of startup companies that are doing lots of sequencing, trying to identify cancer, work in cancer, and much less in infectious diseases, but more in terms of human, which what you guys are doing. Yeah? Okay. So there was a pilot study in 2012 we were involved in the Olympics. And they said, okay, what happens if there was an outbreak during the Olympics? What if, which happened, by the way, in the last Olympics, didn't it? You remember, I think it was norovirus, wasn't it? Yeah, I think it was norovirus. And one of the athletes, he went in and ran by himself or something. You don't know what I'm talking about, do you? Okay. So what happens if there's an outbreak? What are we going to do? So they said, okay, there is something called the whole genome sequencing. This at that time, 2012, and they said, okay, why don't we trial this out? Let's do a pilot study. So they ran. MySeq is an instrument, Illumina instrument. It's called a MySeq. It ran for total DNA extraction of a stool sample. And they did basically, I don't want to sort of go into details into de novo assemblies. You know what de novo assembly is? Yeah. So basically they did a de novo assembly of reads, they sequenced it, and then they brought the reads and they tried to join the reads together. Yeah, without a reference genome. They didn't have a reference genome, they joined the reads together. Resulted in contigs. Contigs are short DNA sequences, yeah, short reads, with significant homology to E. coli 0157 genomes. Okay, so they took that stool sample and they sequenced it and they found, wow, amazing, it's very close to E. coli, it's got lots of E. coli in there. And then they did this typing where they did a SNP-based approach. What does SNP stand for? Good. Sorry, I don't mean to insult anyone in terms of your intelligence, but I don't know what you've known and what you don't know. SNP-based typing of isolates, grouped them according to traditional typing, but then they found quite a lot of discriminatory power, and we'll show what, how whole genome sequencing has helped in terms of typing. Okay. Any questions? No. Anyone asleep? Yes. Okay. I will give you, well, I'll give myself actually a break. Say at 3 o'clock. Is that okay? Maybe for like 5 minutes, 10 minutes. Yeah? But I'll put a condition to that. If you don't respond and talk, I won't give you a break. So, and not just from one person. You see, the people in the front seem to talk. The people at the back, God knows what they're doing. You just don't know what you guys are doing at the back. For all I know, you're smoking and Okay. Challenges of applying this to routine microbiology. Okay. So important considerations. You've got to take species by species with evidence base. All right. So what we're talking about here is what are the challenges for whole genome sequencing in routine microbiology? Yeah. You've got to focus on species by species, and you've got to provide evidence into how you came about in identifying that particular species. Multi-purpose characterization, so you characterize the species, but it's got to have a, the whole genome sequencing will give you multiple purposes, right? So, for example, it will identify the species. It will also give you antibiotic resistance profile. It will also give you a typing. Why? Because it's got the whole genome at its disposal, right? You've got the whole thing right in front of you. You can do whatever you want with it. Whereas before PCR, you've just got a specific gene sequence, and you don't know if that gene is linked to another gene, it's related to another gene, etc. Right? But here you have everything right in front of you. And then you can, of course, find out the diversity within that particular genome and inter and intra-species relations. Mixed model for sequencing and bioinformatics, quality and accreditation, results for both clinical and public health purposes, all self-explanatory. So some background studies. The thing is that once you use whole genome sequencing, you do need some thorough validation. You can't just present the results without any thorough validations. You've got to ensure correlation with existing methodology, although the current methodology may be a poor gold standard. Another thing is that you can determine the variation characteristics of a species. You should find out whether in technical replicates, whether when you keep on sequencing the same strain, the same species, whether you get the same results or not, right? Because sometimes you do exactly the same thing, keep on repeating the same thing, but you don't get the same sequences out. You got to find out if that, then there is a problem with your sequencer, there's a problem with your techniques. Remember, this is all you're trying to validate your methodology. Within a patient at one point in time, right, you've got to find out, you take a sequence, you take a sample from a patient at one particular time, you sequence it, and then you take it again at that particular time, and then you find out the difference. Within a patient over time as well, you take it at different times. Within an outbreak, within the population, and relative contributions of recombination. We'll talk about recombination in a bit. So species by species, is there a good use case? The traditional ways to try to do certain typing mechanisms and identify bacteria, right? The question for all these senior scientist is that is it cost effective to replace what I'm doing now with whole genome sequencing? That's the question, right? We've all heard about how amazing WGS is, but is it cost effective or not? I mean, why would I pay more? Okay, I'm going to get more, but money is tight these days. Okay, so perhaps an example is a replacement of salmonella serotyping. Right? Is it cost effective? Because at the moment they do it all in the traditional ways, but is whole genome going to be cost effective? What about speed? Would it give an added value? Multiple outputs from one particular test. So for example, as we mentioned, you get identification, you get typing, you get AMR resistance, etc. Would it give extra resolution, increased discriminatory power over traditional techniques? So all these are questions that certain people have tried to answer, and they say, yes, it does give more discriminatory power. Yes, it is cost effective. Yes, it is faster. Yes, it does give you more added value, right? And people have published many papers to try to prove that. In terms of quality, remember what we're talking about is making sure that these sequences are of good quality type in terms of you don't find many errors in the sequences themselves. Results, you want to produce certain results, not just raw data. So if a clinician comes to me and says, well, I've got these 10 samples, give me some information. I'm not going to go back and give him sequences. Like I give him a USB full of all sequences. He wouldn't know what these mean. They're just all AGCTs, okay? I've got to give him results. I've got to give him what type of MLSD is. That's the role of a bioinformatician. You've got to take all these raw sequences, huge amounts of data, all these alphabets in front of you, right? And you got to transfer them to results, meaningful results, typing, identification, serotyping, and all these stuff like that. And that's the challenge. Technical and clinical validations, you've got to validate it in terms of bioinformatics, and you've got to validate it in terms of the clinician. If you provide the results to a clinician, he says, what the hell is all this? This doesn't make sense to me. I had an assumption that the sample I've given you is a Streptococcus pyogenes, and you've given me an E. coli. That doesn't make sense, right? So the clinician has to validate the results. Requirement for the whole genome sequencing equivalent of traditional lab controls. Again, that's very important. Each pipeline component designed to output quality metrics, and mapping-based rather than pure de novo. Mapping-based is where you have a reference. And remember we talked about GenBank? You have a reference where you compare your genome with it. Any question? No. Okay. Quality-based components. So for example, if you develop a software that does something, you give it a sequence, it does something magical, and then you get something out. Yeah, you've got to make sure that you provide certain matrices. So a matrix is reported. You give the coverage, minimum depth, maximum percentage of non-consensus bases. Am I making sense here? Probably not. Do you guys know what coverage is? Minimum depth? Yes? Good. I can see some nodding.
Who validates the software?
Sorry?
Who validates the software?
So we have to have tests. That's a very good question. So what we do is when we develop a software, right, there are certain techniques to try to test the software. So testing the software is vital. And you've got to test it with certain other data, not the data that you've been trying to always play around with.
But is that done nationally? Or is it just
No. There isn't like a... I mean, there is accreditation, but this is all very new stuff. So we're not talking about general software development. We're talking about whole genome sequencing data analysis, yes? Or are you talking about general computer science software?
Like everyone is using the same software and they
Yeah.
know exactly what it did. It would be
Sure. I mean, the thing is that there isn't a general body that does that. You've got to, I mean, people rely on you testing your own software and finding out if it's good or not. We've published papers, many people have published papers saying my software is the best, my tool is the best, and this is why. Okay, and I've tested it with this, and that data. And usually what people do is either they sequence their own data, their own samples, or they get stuff from GenBank, and they start testing their software based on that. And they say, yeah, I've got this accuracy, and these are the matrices, this is the coverage I got, well, whatever.
So there are no standards.
No, there's no standard. Okay? So software to discover presence of genes, this is vital because if I'm interested in like a list of, say, 10 genes that are toxin genes, for example, okay? And I'm telling you, okay, I've got these 100 bacterial samples. I want to give it to you. Let me know if these toxin genes are present in these 100 bacterial genomes. Yeah? So I've got to whole genome sequence it. I've got to make sure I develop a software or use existing software to try to find out if these 10 genes are present or not, and with accuracy. And I have to provide the accuracy. So metrics reported depth compared to control single copy gene. Is there a mixed base? Maybe some of the bacterial samples you have given me are actually not the bacteria you think they are. Maybe it's a mixed sample. Okay? It's the sample you have provided me maybe is half E. coli, half Streptococcus pyogenes. There's been a mixture in the lab maybe, okay, where someone's asleep, like some of you, who puts in injects like a DNA of E. coli and then puts something else in there and then presents that. We've had many of that, where we give in samples and we find out in our identification system that it's a mixture. And you say someone screwed up somewhere. Or it could be a genuine mixture. It could be a genuine mixture that someone has been infected with a mixture of viruses or bacteria. And that makes life, obviously, much more difficult. So centralized bioinformatics analysis, a single point for maintenance of complex hardware. You know, this is administrations of infrastructure. We have huge clusters. You guys know what clusters are in computer sense? Yeah, clusters of computers. Basically, it's just like lots of stacks of computers, right? Powerful computers. Because the thing is that you've got so much data. Where are you going to put all this data? Right? You got to put it somewhere. You got to store it somewhere, surely. And so, you've got to buy all these storage. And now everyone's talking about putting them in the cloud, and then it just goes up there in the cloud, and God knows what happens up there. And the thing is that in Public Health England we have paranoia. Unfortunately, we have these like old scientists. I don't want to be ageist, but they are old, and they just don't like sharing data, and I think that's just so stupid. I know this is recorded, and this is a message for you, yeah, if you're watching it. It's just they don't like sharing data because they generate all this data and they say it's mine. It's like a baby, a child says this toy is mine. They say no, you got to share. Sharing is good. They say no, it's mine. And they do the same thing. They say mine, that's mine, my data. You say no, you got to share it. You got to give other people opportunity to analyze it. You say no, that's mine. So, you've got some people like that, which makes, it basically affects the progress of science, I think. But what we have now is we have a good move towards making data available publicly. Anyone heard of ENA? EBI? EBI, okay, European Bioinformatics Institute. And EBI have a repository called ENA, okay, European Nucleotide Archive, which basically provides public, it stores data, and it provides the data publicly for people to access. Okay. Any questions? Anyone wants a break now? Yeah, you're clever. You don't want to put your hand up because then I'll think you're bored. Oh,
Go on.
yes.
So this is, forgive me for asking
Yes, sure.
probably an obvious question, but what is your, in the best wish scenario, what would your shortest timeline be? That's the first question.
Shortest timeline for?
For whole genome sequencing of all microbes.
Okay, I'm about to answer that question in the next few slides, but go on.
And then the next one is, what's your predictive accuracy for whole genome sequencing for antimicrobial sensitivity, in other words?
A good question. I'll answer the second one first, because then I'll go into the first one in a bit. The second one is actually interesting you've asked, because I'm actually doing a study at the moment where I'm comparing lots of antimicrobial resistance tools, bioinformatics tools, and finding out and comparing them with phenotypic data. See, the thing is that at the moment, it takes around 3 days. As I said with my own daughter, it took around 3 days for the results to come back, because they grew the bacteria, then they went, then did all sorts of things, and then they gave us a detailed results into this antibiotic is resistant, this antibiotic is susceptible, etc. Right? What bioinformatics has done is develop tools and databases to try to do whole genome sequencing on these particular bacteria, right? And then generate the results straight away. But the thing is that they have failed quite a lot in terms of accuracy, right? The moment, I haven't published this, but what I have found an average of around 80% accuracy compared to phenotype. So phenotypes is obviously what we think is 100%, right? There is a debate about whether phenotypic results are 100% right or not. But let's just assume phenotypic results, the report that they gave me for my daughter, it says that this particular antibiotic is resistant, etc. I'm going to accept that 100%. But the tool, if I was to add that put that data into my tool, or not mine, but the tools available, what I have found is that they're around 80% accuracy, which is not good enough, because you don't want to come back and say, look, this antibiotic is sensitive, whereas it's resistant, and you start treating someone and they're not becoming better. So we've got a long way to go to try to develop tools that are 100% matching phenotypic data. Yes?
If I'm correct, you wouldn't necessarily need to get 100% accuracy as long as it was better than what's already. I was just saying, I saw them with what we suspected was a UTI.
Yeah.
And in our heads, we're thinking, there's an 80% chance it's C. coli, 10% chance it's Proteus, 10% chance it's something else.
Yeah. Absolutely, but you still want to narrow it down at a faster pace, don't you? There are. If I was to give you 70%, 30%, etc., you still wouldn't really know which. You'll have an idea which antibiotics to give, but it still won't be for certain cases with the rise of antibiotic resistance and stuff. You do need much more accuracy. But I agree with you. But I think it's a good goal to reach, to try to get to 100%. And people are working on that. They still haven't got there. I don't know if I've answered your question in the back. Kind of? Yeah. The answer to the first question I'll get into in a bit. That's all right. Okay. We're going to go through this, and then we'll stop for a break, and then we can continue after that. Okay, this looks very complicated, but this is basically the whole genome sequencing pipeline of PHE. And all I'm going to do is I'm going to go through these steps quite quickly, just to give you an overview of what our pipelines do. And this is all fully automated, so we don't have any sort of interaction with it. It just runs completely by itself. Okay? So that's the first thing. The first thing we do is, well, it's bacteria or virus, whatever, you culture it, and you extract the DNA, and you submit it to you put a 96-well plate, and you submit it to a system, a well-based system, where you select which particular bacteria, this is the lab technician, which particular bacteria they think it is. So if the hospital have said, yeah, we think this is an E. coli or salmonella or whatever, then he or she will put salmonella in the system, right? But I'm not being sexist to put a woman here. It could be a man as well. You never know, you get in trouble these days, just to say. Sample preparation, 24 hours it takes. So you grow the bacteria, and it takes around 24 hours to do that. Next this is all, of course, in the lab. You have all these robots. They do sample dilution, library preparation, and they submit it to another system called LIMS, which is a laboratory informatics system. And they registers all that information there. And then what they do is they start the sequencing process. So this is the sequencing machine, right? The Illumina, what we use in PHE is Illumina MySeq or HiSeq 2500, the HiSeq 2500, rapid mode. So the sequence technician loads the plates into here, and overall library preparation and sequencing takes 72 hours. So that's 72 hours plus 24. It takes a long time, and that's one of the drawbacks. So then what happens is when this finishes, we have huge grid engines. This is a Unilever grid engine, computer clusters, which stores the data, the raw data that is being provided from the sequence. And then there's another high performance system called Lustre, which basically then is taken by the Unix administrator. This is administrated by, you guys know what Unix is, yes? Another operating system put into to Windows, which if you're going to become a bioinformatician, you have to use Unix. You can't be a bioinformatician who uses Windows. That's just criminal. So you got to use Unix, and then there you get the sequences. This is the raw sequences. And there's something called deplexing, which it basically converts the format, the original format from the machine into raw sequences, which that takes around 4 hours. So then you get all your AGCTs from that particular sample. So then what happens is the bioinformatics job starts. So all that, before that, is not really bioinformatics. All that was the lab sequencing and then the Unix admins and all that. Now the bioinformatics pipeline starts. So you've got a bioinformatician here, nice haircut, and then you've got the first thing we do is we do trimming. So basically we trim the either, they call the adapters and either end of the sequences, the reads. And basically we increase the quality. So if there is a part of the sequence that has bad quality, we remove that, okay, and keep the good quality sequences, okay? There is some values, something called the Fred score, that you assess whether a good sequence, whether to keep a good sequence or not. Oops, no, that's not what I wanted to do. That's what I wanted to do, okay? And then what you do is we do what is called a k-mer identification, where we have these substrings. You know what k-mers are? K-mers are these substrings. And you choose a particular value for these substrings, and then you take all these and you create a database of k-mers of a particular length. And then you take these sequences, the raw sequence here, and then you compare it with the k-mer identification database. And then what happens is, what I'm going to explain after the break is a typing mechanism called MLST, multiple locus sequence tag. And then it all depends on which particular workflow. Remember at the start you chose a workflow, what we call a bacteria or workflow, yeah? If you chose Streptococcus pyogenes or you chose Salmonella, here the whole system will trace back from the start. It knows which ones you have chosen, so it goes into the right path, the path of that particular workflow, what you actually require from that. So do you require MLST? Do you require serotyping? Do you require gene profiling? Or is it actually a virus where you look at the consensus sequence and then you look at drug resistance, for example? Okay, so we have all these paths available and we keep on logging every single step. So we know if the error is out, we have everything ready. Okay, and the final thing is what we do is, so we provide a PDF report of all the results that have been generated. Remember, this is from raw sequences all the way to giving you some sort of something to work on. So we either give it back to the lab scientist that has already started this and epidemiologists who have extra research work on this, either give it to Department of Health officials that might be interested in an outbreak, or give it back to certain doctors who also have they have a question. They've asked a question, and then we're answering that question by giving them that PDF report, which has detailed information about that particular organism they submitted. So it went through sequencing and then through data analysis, and then it gives them all the information they require. Okay, that's it. So that's the whole workflow. Starts from here, goes all the way to there, and then to here, and then all the way back to here. That's the workflow. It goes like that. Yeah? Is that okay? Okay. Oh, it's exactly 3 o'clock. Can I give you 10 minutes? But if you be back here by like 8 past, so we can start at 10 past. Sure.
Planning out the project, which is probably going to be a mostly literature-based one. But basically, I'm really interested in, I'm an eye doctor.
Okay.
And I'm really interested in the microbiome of the eyelid and conjunctiva. I'm hoping I can work out a tailored question for a systematic review and also get a lab project and also to do some pilot samples just to see if we can get data. Because at the moment, what we do, literally, to get samples from the eyelid, you can just scrape it. But when I spoke to Dr. Cox about this, who's in Rams here, he was really worried about the low biomass, because I never really thought of it. I just kind of assumed you extract DNA directly from these little cotton-tipped swabs. But I need to read the papers again. But what you were saying about the definitions of different microbes. I mean, this is an area of really evolving, if I've understood it. And what you were saying about the phenotyping, because perhaps based on using the antibiotic and seeing what it's killed in the lab, right? And that's taken as the gold standard.
So, what's the what is this a proposal you're trying to make?
This is a proposal I'm trying to work on. I'm trying to make it contain. It's only got 7,000 words, and ultimately, so the reason I'm interested in it, so infections in the eye can be really serious, right? And even if they're not, they can cause lymph morbidity. We're treating empirically at the moment. So that there are kind of the really serious ones, and then there's the less serious ones. So for contact lens wearers,
Is it Pseudomonas?
exactly.
So we did work on Pseudomonas as well. And we sequenced. That was around 3 years. We published as well around 3 or 4 years ago. So there was a particular, again, an outbreak of some kind in an area, and we had to sequence it and find out. That was the start of our sequencing life. I don't think so. No. Sorry. Go on. Yeah.
So you don't know anyone who's doing anything related in this area? So I'm trying to think who
Not really.
can help me tailor this. So in order to, bark up along the tree too much. But because the other thing is you can get inflammation of the eyelid, which is called blepharitis, and people with blepharitis, we know clinically, are more at risk of getting contact lens-related infections. So what I thought would be really neat is if you could somehow, and at the moment it's just a theory, like we don't even know, because no one's really looked at the microbial load on the eyelid, but if we could measure it in some way, and then we could have a system to test all the therapies we're currently using to reduce the microbial load on the eyelid, and also hopefully be able to counsel patients appropriately about whether they're going to be a high-risk contact.
So what's the problem obviously you have patients and you can take swabs and samples, right, from them?
This is part of the problem. There are basically, so dry eye is a problem, and contact lens-related infections are a problem. And they're probably related because we know people who have blepharitis are more likely to have dry eye, and they're also more likely to get these contact lens-related infections. And at the moment, basically, people buy contact lenses and often not really advised around the risks of wearing them, which are much higher than people who have blepharitis. And sometimes they realise and they just don't wear them. Sometimes and then they get all these sometimes devastating infections. We see tens of patients a day with all these infections, so we give them these intensive drops as tiredness from work. So this is why I'm interested in the question. I'm trying to condense it into a suitable proposal. And with the systematic review, what we normally do with the systematic review is have an intervention, but it's all at it's not quite there yet at that stage. And basically, do you have any suggestions about how I can take it?
So, I mean, it all depends on whether you have access to sequencing services or not. Okay. Yeah. I mean, the thing is, that there is which I'm not going to mention today, there is a very new approach, or something that a lot of people are working on, which is called metagenomics. A metagenomics approach, where basically you take a particular sample and you look at the multiple, you analyze the multiple species, multiple bacteria in there, right? And you identify the multiple bacteria that are in that particular sample. I just wanted to know what is it you wanted to achieve at the end of this project? What is the final?
So ultimately, a good market if I can also get a publication out of it, and what interests me ultimately is being able to work this into a study. But to do a study of patients, we need some primary data. We need to show that we exhibit all the different.
Have you looked at anyone who's done some work on trying to use whole genome sequencing on taking eye samples?
Yes, and it has been done, but they're doing really badly. Like they're tiny patient samples, they're not defining their patient groups well. They're all kind of very early proof of concept.
So I mean, the thing is, I will recommend. I don't know, you're going to have a bit more thought about this, but I definitely recommend something along the lines of using metagenomics for that, and taking the critical thing about using whole genome sequencing, you've got to have many samples. You can't take one or two samples and then base your analysis on it. If you have, say, around 100, 200 samples, and you can collect the data from that, and then you sequence all this information and look at the microbe of the whole genome, identify which particular bacteria are and then perhaps just by that, that's publishable. You look at the number of this particular bacteria that are always appearing in it.
This is it. So the old culture techniques, you get like 2 or 3, but when they've done the whole genome sequencing, they've got like 60 different bacteria, and because their samples are less than 30 in all the studies I've read anyway, that it's how could we do this?
Yeah, I mean that's what metagenomics does. It gives you it gives you a whole sort of what percentage of each bacteria are present in that particular sample. But the thing is that what you can do, if you see a particular pattern, you can delve into these samples and look for certain markers, look for certain genes that are perhaps causing this. You can look at toxins, you can look at bacteriophages, you can look at these particular, you can delve, the thing is about whole genome sequences, you can delve into the whole genome. You can fish around for something that you can see as consistent and constantly available throughout this, and perhaps something within these genomes is causing this. So you've got to delve into the genomes, if you know what I mean.
And when you say 100, 200 samples, are we talking about from different individuals, or could it be from an individual at a time?
No. Well, you can do that, but that wouldn't really give you a variety. You know, you want some diversity within your. You can do that, of course, and that's a good thing if you do. But you really want to know if this particular bacteria or these few bacteria seem to be prevalent amongst many different patients, and then so it gives it a bit more importance. It gives it a bit more importance, and then you want to verify or validate your findings. If you find certain genes that are known to be toxins and stuff that are available and constantly they're associated, then the more samples you have, the more validation.
Understood. And this field is moving so fast.
It is, yeah.
Do you think it's almost a silly thing to try and do a review on because it's going to be out of date, or is that just? I think,
but you've got to. I mean, I don't know. You want to do it specifically on eyes?
Yeah.
Yeah. I mean, it's a very good. I mean, it will be interesting only for those people like yourself, right? I mean, it's not as a. But the thing, I know what people in Sangha are doing. There's a new company in Sangha. Which is basically it's a multi-million pounds project. Well, basically it's taking stool samples from so many different patients and doing metagenomics on that and trying to find out. I mean, they haven't published anything. This is all. Yeah, they're trying to find out the various bacteria that are involved in different diseases and things like that, all directly from stool samples. So you can do something very similar, where you can concentrate on the eye and take the. I don't know in terms of how feasible it is for you to get around 100, 200 samples. I mean, that's the thing.
Not for MSc project.
Yeah.
In the grand scheme of things, getting the PhD is such a common topic.
Yeah.
Actually, and we're doing another study in genomics. We're doing another study looking at this patient group. So actually, I'm thinking something in the grand scheme to get samples. It's just trying to get food into this.
Yeah. But you do need some bioinformatics on that. I mean, if you're gonna put, this is for your MSc project, right?
Yeah.
So you can feel confident you can do some bioinformatics on. Yeah. So that's the thing. I mean, because this is your project, you need to show that you have done this work.
Which is why I was thinking I could concentrate on my strengths, which understand the clinical side.
Yeah.
But I clearly have to demonstrate enough genomic aspects.
Yeah.
But
if you want, let's. But the thing is that you've got to. I mean, if you sequence them, perhaps this could be something that we can help you with. Well, it all depends on the politics and all that. But let's keep in touch by email.
great.
Let me write my. Yeah. That's my name. Yeah. Yeah. Oh, okay. Okay, thank you. Okay. All right. Okay. So this is just very quickly on slides into how bioinformaticians like to work. This is specifically on if you're interested in bioinformatics. So basically what we do is all the data that, this is the raw data that comes out. Anyone knows what this label is for? Which? Linux, yes. Ubuntu, Linux, yes. So we usually use all the Ubuntu, Linux, programming, etc. Anyone wants to be a bioinformatician here? Maybe? Maybe? Anyone likes programming? One. And what do you think? You see, the thing is, I'll tell you of my story. I did a genetics degree, and then I did a PhD in bioinformatics, which is, at the start, it was very difficult. Programming was very difficult. But the thing is that programming is so powerful that once you know it, right, and you know how to use, how to script, even a simple program, then your life will become so much easier. You know, especially if you're using data, if you're using lots of data and stuff, your life will become extremely easier because you'll think that this is something that I thought about, you know, I couldn't really think about doing in a few minutes. Whereas if you write a script, it can be done in like 30 seconds or even less, right? So then you start to really enjoy it. At the start, it will be difficult, but then you start to really, really enjoy it. And I highly recommend it. I do recommend that. There are lots of online courses at the moment, Python courses. So the most the language that is used in bioinformatics now is called Python. And just type Python training or whatever courses or whatever online. You've got to know Unix. So these are two things, Unix and Python. Yeah, and it is well worth knowing, and it's a skill that you can add to your CV even if you don't become a bioinformatician. I think it's something good that I know doctors and people who got nothing to do with bioinformatics, they don't work in bioinformatics, but they've actually got into this and they've trained themselves to at least do the simple stuff. But I tell you, this is definitely the future. I mean, I've been involved in this for many, many years, and now this is really getting very, very sort of busy with lots of data. This is, you know, if you put in your LinkedIn profile big data, then a lot of people will come and chase after you. This is what is called big data now. They want people to analyze big data, mostly artificial intelligence, machine learning, data mining, how to actually try to make sense of the data. Yeah, these are the new biggest things. So I do recommend it if you have an interest in that. Okay, I'll just forget that. All right. So there's a difference between software and scripts. So I can write a script, say "Hello World," okay, and it's just like one line, print, hello world, right? That's a script, okay, which then when I execute it, it comes out, prints hello world. But that's not a software. I can't go and boast about I wrote a software. Software is much more complicated than that. It actually, there is an input and there's an output and it actually does something. Software is endpoint focused, it's transparent, quality assured, tested and validated, it's documented, locked down, accredited, all these fancy words. Okay, so you know the stuff that comes out from the bioinformatics pipeline, all the blah blah stuff. Okay, these are the analysis. You know, the thing is that we can't provide this to a lab scientist, doctor, epidemiologist. They're not really human readable. So therefore, we have to also make sure that we provide human readable stuff. And also, what I mean by that is there are now software and tools developed to look at phylogenetics trees, and they are linked to maps, so Google Maps. So they sort of, for example, Ebola virus. Okay, it traces where it started from, all right, and then it shows to you in terms of a map, shows you where the patients have actually traveled, so where the infection has spread, And also the half of the screen shows you that, the other half shows you a phylogenetic tree where how all these particular species are related, all these samples are related. Are they related in one clade or are they not relating? This is sort of what human readable means. You can, a doctor or lab scientist or whatever can interact with these things and can say, well, this is interesting, especially if you can play around with the tree, play around with the, I mean, I don't have anything here, but we do have something called MicroReact, which someone called David Arnensen from Imperial who's developed. It's a very cool tool. But it's obviously specialized for people who are specialized in different bacteria. Just very quickly in terms of identifications, you know, not all samples submitted are what they say they are. So remember when I told you the lab technician said, yeah, submit it, this is salmonella, but not all of them are, you know, they think it's salmonella, but eventually it turns out to be not salmonella. So not all samples are pure. And they did sort of a quick study in PHE, and they did 53 samples from a range of species, sequenced Illumina, and these were the cast, E. coli, Legionella, Listeria, and all the Boletti, and if you can read all those. And different salmonellas, Shigella, Streptococcus aureus, and so therefore they did a species identification, and they found out that with our KMA tool, we have a KMA tool, as I explained before, it reported as the distance to the closest GenBank prokaryotic genome match. So it gave 39 out of 53 gave a close match, so a distance less than 5, the correct species. 10 out of 53 gave an intermediate match, and 4 out of 3 gave a distance match. So, and then they say the cause of each case of misidentification was the lack of close genome with GenBank. And that is the problem here, that because we rely a lot on GenBank, we rely on what GenBank has already identified as species, and we populate our database based on GenBank, then anything that we don't get, we tend to, you know, if we don't get a close match and we get a distance match, then it's probably that we don't have a closest reference in GenBank. And GenBank is continuously, they're populating it more and more and more. And hopefully in the future we'll have majority of the species being represented in GenBank. So typing, this is very quickly, I'll just talk to you about MLST. And MLST typing using whole genome sequencing. So, anyone knows about MLST? No? Anyone heard of MLST? No? Okay. So it's multiple locus sequence tags. So this is a typing mechanism, yeah? If I want, remember we talked about typing, basically giving a label to a bug, right? So if I have salmonella, there's so many different salmonellas, I want to label the different types of salmonellas, okay? So I'm going to do a typing mechanism. How do I do it? I'm going to do the following. So I sequence a whole genome. I have many reads. These are short reads, yeah, short isolates. I map the reads to an allele sequences. So these are what is known as housekeeping genes. So these genes are present in every species. Yeah? They are present in every species, these particular 7 genes. But the thing they differ is the certain SNPs and certain mutations in them that makes them different. Yeah? So we look at these 7 genes in whatever species it is. I look at these 7 genes and I say, if the allele in this AROC gene is this, then it's that type. Because we have a database where we know every single allele belongs to which particular gene. Okay, so the first thing I do is I map my reads to all the allele sequences, yeah? And then I get all the aligned sequences with the genes. And then, so here you can see that, so for example, if we take just the first gene, AROC, If the read sets match to 100% to the allele sequence with 0 SNP indels, extract the allele number. So I go to, in my database, I have a gene called AROC1, which has allele 1, which has this particular pattern, all right? My sample does not match this. It does not match that. What it matches, it matches AROC3, which matches exactly 100% the sequence, yeah, for that particular gene, for the allele AROC3. And I do the same thing for all the other genes. I do the same thing for DNAN, HAEMD, and all the others, okay? And at the end, what I get is what is called allele variance. So remember AROC3, I get number 3, the variant number 3. And I do the same thing for DNA. I got my gene matches variant number 1. For this, it matches variant number 2, matches 1, 3, 3. So then I got a profile. It's called a profile, where my MLST profile is called 3121331. That's my profile for my sample. Is that okay? You guys understand that? So then how do I determine which sequence type it is? What is the typing? I go back to the database and I ask the database, do you have any profile that has a 3121331? Do you have it or no? And the database says, yes, I have it. The sequence type 7 has a 3121331. So my sample has a sequence type 7. So I report back to the doctor, I report back to the lab scientist, as that sample has a sequence type, a sequence tag of what? Of 7 MLST. Okay? So then they'll identify. So if you have an outbreak and all your samples have an MLST of 7, you know that there is one specific type that we should really be worried about. But if you have multiple types, if you have 7 and 19 and 360 and whatever, there's so many hundreds of types, right? Then you know you have a population which is mixed, different types. Yes, they all are salmonellas, that's fine, we know that. But it's not one particular type that we should be worried about. It seems like a whole mixed population. Yeah? Any questions?
Within that one type, you could still have, because one of the papers was given, you could still have, for instance,
Absolutely, yeah. You can have various populations, yeah, absolutely. But the thing is that in these particular 7 housekeeping genes, people are more confident to say that if you look at these 7 housekeeping genes, they label that particular sample with that. But there are other genes. Is that what you're saying? There are other genes with different profiles.
well, no, I think what I was trying to say was you're only looking at a distinct population there, it's type. So you've got a cohort, haven't you? That's not representing the entire genome. So
Yeah.
you wouldn't pick it up.
No, you wouldn't. You're absolutely right. You're absolutely not. But that's not really generally. I don't know what you said. You've read a paper on this, but it's not generally the case. It's not a typical case. Usually, they look at these. It's not usually that particular gene is important, but it's usually they look at these 7 genes and they type the sample according to those 7 genes. But if it is important, then of course you're not taking that into consideration. You're just looking at these 7 housekeeping genes, and that obviously can change the type. But I think there are rare cases. I'm not sure. What does the paper say? Does it say it's common or?
It was just an example of an outbreak, MRSA.
Yeah.
We were given to look at.
Okay. Yeah. I guess in outbreaks, yes. Any other questions? Okay. So the thing is, they look at the confidence matrices of the type, and they say that so that we can be confident this is the right gene and the right information we have taken, they label different types of conditions to give to the MLST type. So you have 3 conditions: minimum consensus depth, maximum percentage non-consensus basis, or percentage coverage. So let's go through each one. So minimum consensus depth, you have, for example, in this AROC C3 gene, right, you take the depth of your genome, of your sample, and you see that it's quite a high depth. You know the difference between coverage and depth? So this is depth. When you go down, you look at in terms of the genome and reads, if you look down, and the more reads you have, the higher the depth you have. In terms of coverage, coverage is this, where you look at this way. Everything has to be covered from this side. Depth is this, the coverage is that. So if you have a really long depth, you are more confident that this is a correct assignment because you've got many reads that have been amplified at that particular region. Okay? Whereas if you have and they label this as green, but if you have a low consensus depth, so if you have like 3 reads here, then they label this as amber. So basically when we report back to the doctor or whoever, we say, yes, we think the MLST is 7, but we give it an amber color because it has low depth. Okay? If we look at the maximum percentage non-consensus basis means that it could be a mixed sample. So if you look at here, you have A's, it's not a consensus, right? There's been a change, there's a SNP. That's what non-consensus means. It means that there's a SNP. And if there's a high maximum percentage, so there's a high number of different alleles within that particular region, then we say, yes, we think it's an MLST7, but it's a red high maximum percentage because it contains alleles that are SNPs, and we're not confident that this is a correct assignment. Here, you can see that there are no SNPs. It's all very clean, 100% coverage, you can see here, and the depth is quite good, so we put it as green, and we say there's a low percentage non-consensus basis. So basically, there's no SNPs in here, in that particular gene. Yes? Does that make sense? Any questions? Okay. There's a very nice tool, if you want to use it, it's called Tablet. Don't just type tablet in Google, you'll get other things, but type tablet genome or something. And it's basically a genome viewer. You can view your reads, you can view your sequences very nicely in a colored way. So here, it sampled with low minimum consensus depth. You can see here, you can, depth of 3. You can see here that it's giving you 1, 2, 3. 3 different, only 3 reads. Whereas here, if you look at here, in this region, you can see that it has a really long depth. Okay? So the thing about tablet is you can see one genome at a time. For bacteria, we have hundreds and thousands. For humans, you probably have 1 or 2. So it should be fine for humans. And I recommend tablet. It's good, very good. If you want to look at specific areas, and if you have a project or whatever, and you can take a snapshot and put it into your project proposals or project dissertation or whatever, I think it will look nice. You say, this is the region of interest. Look, it has high depth, it has good coverage, and it doesn't look like it's a mixed sequence. It looks like there is a clear sequence. Okay? Percentage non-consensus basis. So to find out what is the percentage of non-consensus basis. So look at this particular position here. And if I want to find out what is the percentage of C's here over T's, yeah, what I do is the number of non-consensus bases. So in this particular case, I have non-consensus basis is 9. So I have 9 C's, 1, 2, 3, 4, 5, 6, 7, 8, 9 C's. And 24 T's. So I do 24 plus 9 is 33. So to find out the percentage of non-consensus bases, to find out the percentage of C's, I do 9 over 33 times 100, which is 27%. So 27% of that particular base here is non-consensus. 27% is C's. Yeah, and so we report back and say 27%. It's actually quite high. Because what we do is we say 15% and over is quite high. You shouldn't really have more than 15%. Okay? So 27% is I mean, 15% is what we say. There is no standard way of doing this. So 15% is quite, is what we have, but anything above that, it's questionable. And we say, yes, this could be a mixed sample. By the way, when you look at humans, this is different, yes? This is homozygous, whereas humans are heterozygous, so it's different. You will have a mixed, this will look different in human population. And the results, this is a PDF results we give. So this is sample name. We just had to hide that out. But the sample name, and then, so here you can see the MLST type. This is a different example, not the example that we've talked about. MLST type, we give back to the doctor, whoever, we say MLST type is 398. This is the profile. Remember how we got the profile here? And then we give all the stuff that I've just given you, the QC mean, consensus depth, percentage maximum, etc. We give the coverage is 100. The coverage has to always be 100. If there is something missing, then we flag it out. And in the report also, we start giving all the colors, the different green and amber and all that. Okay. So there is just to give you an example of an outbreak. So how typing has helped in terms of outbreak investigations. In France, there was posted, there was a European alert. There was an increase in this particular type called PT14B, and had an egg isolate with German egg mark having the same MLVA and SLV profile. So these are two different typing schemes for Salmonella. Okay. There were cases in Austria, Luxembourg, and Germany with the same SLV. So that's how they found out. They typed them all, and they found out that the cases in these three countries had the same type. Trace back investigations led to sampling of egg production premises in Germany. They went back and found out where did these eggs come from. They all came back from the same place in Germany. Company had 3 separate egg production plants with layer flocks from a single supplier in Germany. The particular type PT14B was isolated from 2 out of 3 sites with the same SLV profile. And they also traced back to the UK as well. So we found back, stores were selling eggs who came back from that particular German production company. All outbreaks involve eggs from the same German supplier. So the outbreaks was Salmonella. It was quite serious for those particular. The PT14B is quite a serious type, and so eggs in France from site 1, eggs in Germany from sites 1 and 2, and eggs in England from site 3. So there were 3 different sites, but the same supplier. And so you can see this phylogenetic tree. I mean, you don't really need to look at the details of this, but to take just the top part, this is all background samples. And you can see here, so you can see here that the black samples, the colored black, are all UK samples. This is all the background samples. These have been isolated from a hospital. It's isolated from some restaurant, sells has noodles, whatever, sell eggs. This is a Chinese food restaurant. It's all been isolated from there. Remember, when you draw a phylogenetic tree to find the relationship between samples, you need some background samples to find out. But here is where all the interesting part happens. What basically you can see here is that the red samples are from the German eggs, right? And you can see here, these are the outbreak samples from the UK, and these are from the French ones. And you can see that they are from one particular source, what do they call this? The doner kebab, that's the one. Yeah, a doner kebab restaurant, which had, was selling eggs and was making doner, I don't know, had some eggs somewhere in there. You can see that they all had, these are different patients, they all had the same SNPs. So they were all very, very similar to each other. In fact, very identical to each other. And it's quite related to the German and the French outbreaks as well. So these are the German samples from Germany, and these are the French samples. Yeah. The reason why it's a bit different is because it's from the different sites. Remember that eggs in France from site 1, eggs in Germany from site 1 and 2, but England is just site 3. So there was some SNP differences between them, but not much. Okay, so that's the SNP differences between these particular isolates, this and that. But essentially, what this shows you is that they found out that they all come from the same source. All these particular isolates, these are the outbreak isolates, they all come from the... And the UK ones are very much related to the German ones. Okay, so that's how you trace back. You say, okay, I found out all these, I've got some German samples, I put it into a phylogenetic tree, I know the typing for them, but look, these are very similar to this outbreak, so therefore they must be from the same source. Is that okay? Yeah? So this is an example of how typing, how phylogenetic, how whole genome sequencing helps in trying to identify sources of an outbreak. Yeah? Good. Quickly, characterization. We have a tool called Gene Finder, where if you've want to know further information about a gene, you want to know information about how to get the antimicrobial resistance information, how to identify the presence of this gene or the absence of this gene, use a Gene Finder. It reads to sequences, it reports the best match, includes quality matrices on depth, high non-consensus basis, and out-of-frame indels. Now, what I'm going to do is finish off by just giving you a nice piece of work that we've done. It's a nice story to how we used whole genome sequencing to identify something that was problematic in the infectious diseases community, and we've published this if anyone wants to read further. So it's a novel tool for early warning of upsurges in invasive group A strep diseases in the UK. So just to give you a background of those who don't know GAS, we call it GAS, group A streps. Streptococcus pyogenes, it's called Lansfield group A streptococcus or GAS. It's a gram-positive bacteria. It causes a spectrum of infections ranging from pharyngitis, mild sore throat to necrotizing fasciitis and toxic shock syndrome. I actually saw it's also known as the flesh-eating disease, right? Certain people have lost all their limbs, hands and legs, because it's just their flesh-eating burns completely. So it's a pretty serious when it becomes, when it enters sterile, when it enters blood, when it's invasive, it becomes quite a serious, when it's non-invasive, it's usually in your sore throat, yeah? You have a sore throat, and if it's bacterial, it's not viral. If it's bacterial, then it's usually Streptococcus pyogenes. It's not serious, it's not invasive, you can use antibiotics to treat it. But if it gets into your bloodstream, then it gets very serious, and this is what is called an invasive. So I'm just going to tell you, here. I'm going to tell you something that happens. So this is, again, group A strep, Streptococcus pyogenes, is also differentiated according to its types, right? Typing. So you've got a gene called EMM gene, right, that characterizes types different group A streps. And so you've got EMM1, EMM3, and EMM89. So these are 3 different EMM types. You can see here from January 2008 all the way to January 2013, the percentage of distribution of these particular types in the UK, in the England population, right, in the English population. What we saw here in this region is an unusual upsurge of that particular type, EMM3. Usually, you see the blue one. Can you see the blue one? This is what is usually available in the population. We usually see an upsurge of EMM1 in the UK population. All right, so everyone's used to EMM1 being quite prevalent, yeah? But then suddenly, for a few months, we saw an upsurge of EMM3 and EMM1 came down. And we wanted to find out why did this happen, right? Why did EMM3, that particular type, just went up and then the EMM1 down, and then they sort of swapped again, and then this EMM3 went back to its original, well, not original, but its normal distribution, and EMM1 back to its normal distribution. Yeah, so that was the question. The question was, don't need to go into these. What made EMM3 quite prevalent? So we used whole genome sequencing to find out why. We had, in fact, we added more, but just to give you the initial work, a total of 295 genomes from different geographical locations within the UK. They were randomly selected from 2001 to 2011. It's very important that if you're investigating an outbreak, you actually give some, as I said before, you give background samples so that you can compare between what happened before and what's happened now. 182 of these were invasive and 113 were non-invasive. So the serious ones were 182. And then what's important about data that we get in PHE is that we have detailed clinical, epidemiological, and microbiological information. So we have lots of information about age, name, obviously, gender, ethnicity, all these information about this particular sample that will help us to find out, you know, how did this particular strain was spread. So what we did was we analyzed the data by mapping to reference genomes, and calling SNPs. Obviously, I'm going through this very quickly. There's lots of work being done here, but this is what is called a phylogenetic tree. Yeah, and you can see three colors here. Yeah, this color belongs to an MLST type. Don't get mixed between EMM type. EMM type is a specific to group A streps. MLST type is what we just talked about. Yeah. So we did MLST for all this, and we found that the whole of the population, our samples, were divided into three MLST types. They're divided into 315, 406, and 15. You can see here, based on SNPs only, this is a SNP tree, right? You can see here that this phylogenetics tree based on SNPs correlates very well with MLST profiles. You can see here that there is no mixtures. All the 315s are here in the bottom of the tree. They're all clustered together. Yeah? You can see here that all the 406s are clustered together. And the 15s are here clustered together, and then there is a few other branches, and 15s are the most prevalent. That's why you have them. But that's interesting because you see that you've got a phylogenetics tree which contains so many hundreds of, we've added more samples other than 295, hundreds of samples, and they've nicely been differentiated into three different MLSTs, right? 315, 406, and 15. Now the question is that why is this box? Why is this being boxed? This particular clade? We found we delved into the genomes of every single sample here, and we found that only majority of these particular samples right here, and not found in anywhere else, contain a bacteriophage. Remember, you know what a bacteriophage is? It's like it's a virus within a bacteria. Yeah, that is not present in any of those samples here. And the majority of these samples here were involved in that particular upsurge in 2008, right? So if you look at, these are the two bacterial phages. This is a phage containing one particular toxin that's called superantigen. It's called SSA, and this phage containing two toxins, SPEC and SPD1. And you can see that they are very similar, except this is showing matching, right? Except these particular 2 genes. This is the phage that is found in here, and this phage right here is found in everywhere else right here. Yes? So there's 2 different phages we found. All these have this, majority of those have this particular phage, and all of these have the one which is called, we call the SSA phage. Okay? But the main difference is between these 2 genes, and these are the 2 toxin genes. And so what we have found is the unusual upsurge in invasive group A streps in early 2009 by the EMM3 could be attributed in large part to the emergence of this novel phage containing clade of EMM3 strains within the population. So for some reason, the EMM3 bacteriophage, the group A, the SPEC one, what we call the SPEC one, this phage, was introduced to the United Kingdom, we found in 2006, right? And for some reason, it became quite prevalent in early 2009, and then it died out. And we believe that the reason why EMM3s outnumbered the EMM1s is because of the introduction of that bacteriophage in the population. Yeah? And that conclusion was only possible when we sequenced the whole genome and we delved into the genome and looked into the bacteriophages, looked at the gene's presence and absence of the genome. We delved inside and found there's a pattern. These samples contain these bacteriophages, whereas these samples don't. And that is the main difference. Okay, and so what? You may ask. So we hope that this work will assist in recognizing the start of a new outbreaks by early detection of novel clades that contain phages. So what we're saying is that if a patient is infected with group A streps, the strain is isolated, you do whole genome sequencing, and you do a SNP phylogeny. And if it's a novel strain that we haven't seen before, then this investigation is you need to investigate further and find out why. And then we will tell the GPs and all the clinicians that look, there is a new strain, there's a new bacteriophage that has been introduced to the United Kingdom, right? And this is an alert where certain people might become even more sick because of the introduction of this new bacteriophage, right? This is sort of the theory that we've come up with based on the findings that we have found. Any questions on that? Okay. This is another example of, this is a different case, but the same species, group A streps. Another example of good concordance between EMM type and SNP profile. So you can find here that all these particular samples are EMM1. You can find huge, very big SNP differences with EMM11 and big differences with EMM90, 89, and 94. So there's a big concordance between AMM typing and SNP profiling. Finally, just a quick comment about sort of the issues with whole genome sequencing and phylogenetic trees in terms of outbreaks. In this particular case, we found there's 400 SNP differences between this sample and these samples, right? But then later on, we found out that by removing the recombinant elements, so you know that bacteria, in terms of the DNA, they go through recombination processes, right? And they continuously recombine. And when they recombine, they exchange DNA and mutations happen through recombination element. When we study evolution, we shouldn't really take these into consideration because you're not looking at the core elements of the genome. You're looking at the changing, the variants, right? You're not giving a true picture. You need to look at the core elements of the genome, the parts of the genomes that don't hardly change. But if you look at the parts of the genome that keep on changing, that doesn't give you a reflection of the evolution of the genome, okay? So when we remove the recombinant elements, you found that is only this sort of shrunk. You can see the difference here. Look, it shrank and became only 8 SNPs from 400, right? So this gives you an importance of, if you're going to draw a phylogenetic tree, if you're going to look at the relationship between bacteria, you've got to find out how recombinant that particular bacteria is. And if it is quite a recombinant bacteria, then you've got to remove the recombinant elements. Finally, like early, there's lots of acknowledgements, all these people who've done a lot of their work. And that's it. Is there any questions? Yes?
Very, very rarely is in clinical practice we come across patients who will die.
Yeah.
They've got an underlying immunological problem, but they're young, fit people for whatever reason. Just
It's not just people. I mean, we're not just looking at people who die. I mean, we're looking at people who get really sick, and they, forgive me for going back to my daughter's example, but my daughter's example was someone who they isolated the samples, and it was E. coli at the end, by the way, but it was a different type of E. coli, it's not a prevalent type, and also they had to look at the antibiotic-resistant profiles and to treat it. So typing is not just for, if you find, those patients with very serious illnesses, but also also surveillance is very important. What we do in PHE is always constant 24-hour surveillance into what is actually going on in the population, what is actually spreading. But you'll be surprised, the data I've been looked at. I mean, I don't know if you're a clinician yourself or not, but in terms of microbiology, a lot of people do die because of infectious diseases. And I've looked at clinical I mean, for the group A strreps, the invasive, I saw the data, we get the data, this patient dead, and some babies, etc. We find quite a few have died and things like that. So it is important. It is, these typing mechanisms definitely help in terms of finding out which particular treatment you should really give. And as I said, it's important for surveillance as well. I mean, the government continuously wants this data. The government continuously wants to monitor the spread of infectious disease, especially people coming from all sorts of places around the world. They bring with them diseases. They bring with them infectious diseases. And especially if it's a new strain, and then if it spreads and it becomes problematic and stuff, so it can be serious. So, yeah. Okay, any other questions? I just hope. I'm sorry if it became a bit detailed and too technical sometimes, but I was told I'm supposed to do this. I hope you found it useful, and good luck with your course. Thank you. Yeah.
If somebody wants to get more into bioinformatics,
Yeah.
doesn't really have that background's genomics and biomedical science, what's the best way to go about it?
Well, as I said, the two things you need to do is you need to try to familiarize yourself with these two, Python programming, Unix. There are plenty of, I mean, I don't know, you're doing this module here, but is there a bioinformatics module? Yeah. So you can, once you finish this master's, you can start applying for jobs. And what they'll do is they do some tests and stuff. If you prove that you can get into, try to write a script to do analyze data or whatever. And then you can start your path from there. There are lots of masters, bioinformatics master's degrees. If you're interested, I know Imperial do one and a few others. Lots of them around these days. There's lots of genomics and all the things. But you come from you'll have a master's in genomics, right? But I think if you can demonstrate some bioinformatics skills without doing another master's in bioinformatics or whatever, I think there's plenty. If you go to jobs.ac.uk and type bioinformatics, hundreds of jobs now. Even for people, they just require a master's. So I think what I'll do if I was you is try it out. Try writing scripts. There's so many online courses. Try to write scripts. One of the biggest things at the moment that a lot of people are looking is R. Have you heard of R? So even commercial companies, nothing to do with bioinformatics, but now banks and everyone's looking for R specialists. I have friends who are economists and stuff. They're begging for people who know R. It's a statistical package, but it still requires programming. So if I was you, I'll get into bioinformatics. Also, really requires R, but it's not a condition. Python is a condition. Python and Unix, these are the two main conditions. So if I was you, I'll start doing some practices, writing some scripts. Maybe it's not for you. Just try it, but maybe it's not for you. Get into it, and if you really like it, practice more and more and more, and then